let's converse about the major histocompatibility complex. No, that's not the one. That's the one. Okay. So as I mentioned, we started to talk about it yesterday. And as I mentioned yesterday, it's pretty much a set of proteins on the surface of the cells that allows your immune system to determine whether the cell is self or foreign. These proteins work in a kind of the same way. What they do, they present the antigen on the cell surface. Consider antigen as the unique signature of the cell. Remember, I use these markers, okay? So each of these markers is the antigen, right? And red marker will be the good antigen, the antigen that's produced by the normal cell in the human body. Does that make sense? It's normal antigen, okay? So if this antigen is presented by the major histocompatibility complex proteins, which let's say it's my hand, this presentation means I'm fine, okay? If the antigen becomes different, that means I'm not fine. I am the bad cell. I am foreign. And we will discuss what your immune system will consider foreign, right? Does that make sense so far? Because we're going to start with very broad definitions and we're going to get into more details, explaining how it works. There are two classes of MHC. Let me call it MHC if you don't mind. Class 1, as soon as the pen starts working, you will see that. Yeah, class 1, which is illustrated in the picture A. Okay? Class 1 presents the antigens that are produced in the cells. And for the simplicity, let's consider antigens proteins. Okay? So it presents short peptides that are produced in the cell. So which cellular process is responsible for synthesis of proteins or peptides? Translation, very good. So normal cellular translation, just normal process in the cell, is synthesis of peptides. Some of those peptides are going to go on the surface and will be presented by the class 1 MHC. Does that make sense, this thing? Okay. Class 1 MHC is present on all nucleated cells in the human body. Which means, which cells do not have MHC1? Red blood cells, and if you consider themselves, platelets. Some people say cells, platelets, not cells. Don't tell anyone. Okay, so all cells, except red blood cells and platelets, have MHC class 1. All of them. Your hepatocytes, your fibroblasts, your immune cells have MHC1. Does that make sense? Are we clear on that? So, think about this. Normal cell produces these antigens, self-antigens, we call them self-antigens, as a part of life cycle. What happens to the cell if it gets infected with an intracellular pathogen, say a virus? What virus is going to do in the cell? In the very basic terms. Why virus infects the cell? To do what? To replicate. Exactly, to replicate. Replication in terms of the virus, what does that mean? You have to produce more viruses. Good. I'm asking really, really simple question. Believe me, nothing tricky. I will tell you if it's tricky. More viruses. Viruses are made of what? Generally. Good. Huh? Protein. Well, capsid is protein inside nucleic acid. Protein. So virus hijacks the cellular translational machinery and produces its own proteins, which inevitably leads to presentation of virus antigen instead of cellular on the cell surface. Does that make sense? Cell starts to produce not cellular antigens, but viral antigens, which look to immune system how? Self or foreign? They look foreign, so they label cell foreign. Does that make sense? Same happens in cancer. In cancer cells, production of those, synthesis of those antigens is skewed, okay? And antigens that are presented on the surface of the cancerous cells are different from normal tissue. That's why your immune system is actually quite effective in recognition of the cancer cells. So, proteins synthesized in the cell, normal cellular markers or intracellular pathogens. That is shown on the picture 1. Okay. Now, class 2 MHC protein, class 2 MHC is present on so-called antigen presenting cells. Now, before we move on to how it is presented, antigen presenting cells, okay? It is a simple question, it contains the answer. What do antigen presenting cells do? Exactly, they present the antigen. Now, they present which two types of immune cells need to recognize the antigen? C and B cells, very good. B cells don't need presentation. They already have receptor. They recognize the antigen directly. Does that make sense? T cells cannot recognize the antigen directly. They have to be presented with an antigen. Does that make sense? Now, the thing is, in biology, when I came to biology, I was coming from chemistry and physics, and for me it was really hard to accept that sometimes you cannot logically make an explanation for something. It is what it is. That's what we observe. So that's what we observe. B cells don't require presentation. T cells do. Okay, that's life. So antigen presenting cells, they present antigen to T cells. How they do it? They phagocytose the microbe. Look at the right picture, the picture B. They phagocytose the microbe, and in the process of phagocytosis, what has happened to a microbe? Bless you. What happens to a microbe? And destroyed, yes, so it's broken down in pieces, okay? Those pieces, parts of the microbe, then serve as antigens bound to class 2 MHC. So function of class 2 MHC is to present the antigen, okay? And in a very simple terms, if this is normal, okay, and this is abnormal, if the abnormal antigen is bound to MHC1 on the cell, what does that mean? Huh? No. So, okay. So this is, I'm a good cell now. Okay, I'm a good cell, I present normal antigen. Now, I get infected, present abnormal antigen. What does that mean about me? I'm sick, I'm foreign, I need to be killed, right? Now, I am antigen presenting cell. So I see a pathogen. That's pathogen. I phagocytose it, break it in pieces, and present it at MHC class 2. Which cells do I present it to? So what's the purpose? I'm kind of leading you. What's the... Not me, because I'm presenting cell. I'm telling T cells, look, this is what the foreign cell looks like. Does that make sense? go and kill everything that bears that antigen. Does that make sense? Antigen-presenting cells essentially sort of teach T cells. They tell them, this is what you're going to search for. Does that make sense? Okay? So it's recognition of self versus non-self. Now think about this. Now again, MHC class 2 found only on antigen-presenting cells, which are dendritic cells, macrophages, and B cells. Now, we've already encountered macrophages, right? That's the component of innate immunity, but turns out it is linked to adaptive. And we've already talked about B cells. Turns out they can phagocytose as well, but for antigen presentation purposes only. They're not major phagocytic cells, okay? They're not. Their function is not to phagocytose, it's slightly different, right? What is dendritic cell? Dendritic cell is a major type of antigen presenting cell. It is kind of a macrophage, kind of sort of macrophage. Does that make sense? But its function not to clean up the debris, not to destroy the pathogens. Its function is to pick up a pathogen, break it in pieces, and teach T cells what to attack. Does that make sense? Now I'm going to ask you at the same time a very simple question. Well, it's very simple actually. The answer is simple. Dendritic cell or macrophage, do they have nucleus? It's a simple question. Do they have nucleus? Yes, they do. Which MHC molecules do they have on the surface? Class 2. And 1? Yes, because there are no exceptions. All nucleated cells have class 1. So they have both class 2 and class 1. Does that make sense? Okay. Now, example of how MHC is important for medicine. Transplantation. My cells produce certain type of MHC molecules that present certain type of antigens. Does that make sense? They're unique. Somebody else's cells produce different, slightly different MHCs that present slightly different antigens. Now, if you put my organ into somebody else's body, their immune system will not recognize my tissue as self. Does that make sense? Because MHC and antigens are different. So that play, and that's why when there's a transplantation, we need to provide immunosuppression. Does that make sense? Moving on. So I wanted to introduce the concept of MHC before we talk about T cells. T cells, they originate in the bone marrow, and then they travel to the thymus. That's an overview of the T cell, the whole T cell process. We're going to talk about maturation in thymus. In thymus, they mature, and they split in two big populations: the CD4 cells and the CD8 cells. And these cells show so-called MHC restriction. Okay, I'm going to write it down. MHC restriction, which means that CD8 cell recognizes sick cells via MHC class 1. So it recognizes MHC class 1 molecules. What does that mean in terms of the spectrum of recognition? CD8 cell, which cells can it recognize as being infected? All nucleated, exactly. It can recognize any cell as being infected. can recognize if hepatocyte is infected or B cell for that matter, or I don't know, hepatocyte or splenocyte, anything. Does that make sense? CD8 cells are killers. Okay. CD4 cells, on the other hand, recognize class 2 MHC molecules. So which cells CD4s will recognize then? Antigen presenting either dendritic or B cells or macrophages. And it turns out that this ability to recognize exclusively MHC class 2 is what defines their function. CD4 cells are helpers. Okay? Now you have to appreciate that T cells do nothing until the antigen is presented to them. Okay? And for the purpose of example, we're going to use dendritic cell as antigen presenting, right? Now look at this. Dendritic cell is antigen presenting cells. Which MHC molecules does it carry? And 1. It's nucleated, 2 and 1, right? So dendritic cell can present antigen via MHC class 1 to CD8s and via MHC class 2 to CD4s. Does that make sense? So it's kind of jack of all trades and actually master of all of them, okay? After the presentation, T cells become activated. They start to differentiate and proliferate. Each of them, both CD4 and CD8, produce set of memory cells, okay, immunological memory. CD4 cells become different types of helper and regulatory cells, and CD8s become so-called cytotoxic lymphocytes that sometimes referred to as CTL, cytotoxic lymphocyte, or just T-killer, okay, T-killer cell. So far, make sense? One thing I just want you to be on the same page about it. T cells have receptors on the surface that recognize MHC molecules, so-called T cell receptors. Does that make sense? You have to appreciate that T cell receptors structurally very similar to antibodies, and they recognize antigens bound to MHC molecules in exactly the same way. And the variety of T cell receptors is ensured by VDJ recombination. It's all the same mechanism. The only difference, T cell receptor doesn't become soluble. You see what I'm trying to say? It doesn't separate itself from the cell, it stays on the membrane. Okay? So T cells do not release it into the bloodstream, it stays on the membrane. Does that make sense? Okay, we're going to overview some steps of that process in more details, right? So first of all, the selection of T cells. Remember, we talked about B cells that mature in the bone marrow. They undergo a certain type of selection. What type? I'm not asking the name. Terms, okay, terms, you will learn terms. Conceptually, what you do not want B cells to do? To attack what? Say again? Your cells. Your cells. You don't want them to attack your own tissues. We good? So negative selection is when you pretty much make sure that all self-reactive cells die. Does that make sense? We good? With T cells, you gotta throw in additional step, which is ability, just general ability, to recognize MHC molecule. If the T cell cannot recognize MHC, whether class 1 or class 2, it's useless because it cannot be presented with an antigen. Does that make sense? So that's positive selection. Positive because only cells that can recognize MHC are selected. Those that cannot go into apoptosis. Does that make sense? I personally call it being fit for the job. Okay? So they just fit for the job. It's like it may not be a politically correct comparison, but imagine that if somebody hired to work as a special forces, people who don't have both legs, you're not going to hire them as the special forces like officer. Okay, they cannot run. Okay, being fit for the job. Now, another thing, second step, is to make sure that T cells do not attack your own tissues. So only those that are not self-reactive survive. So that is negative selection, same as with B cells. I call it cells do not go berserk. Right? So the final pool of the cells must be able to recognize foreign antigens bound to MHC, but not your antigens bound to MHC. Good? Okay, that's really a foundational concept that then will help us to understand how these cells work. So first, let's talk about, okay, we got them produced in bone marrow, we just got them matured in thymus. We have a pool of competent T cells. Where do they go from thymus? Lymphatic system, they stay in the spleen or lymph nodes. And in the lymph nodes, are they getting directly exposed to the antigen? No, not directly. What should happen? Can they directly recognize the antigen? No. They have to be what? Presented. So antigen gets into the lymphatic system. Dendritic cells or macrophages, well, mostly dendritic cells, will phagocytose an antigen, break it in pieces, present it on the surface, right? So here's CD4. CD4 cell, shown here as the blue cell with a purple nucleus, okay, via the T cell receptor, recognizes MHC class 2 on the surface of antigen presenting cell, that sea urchin looking like dendritic cell. And that leads to activation of CD4, formation of the pool of memory cells and pool of helper cells, right? This process, the differentiation and proliferation, as we mentioned before, is called clonal expansion. Okay, the clone expands. And function of CD4 cells, which we're going to get into more details, is not to kill pathogenic cells. It is to help other cells, like B cells or macrophages, to develop their abilities. Okay? So let's take a look how they do it. We've got CD4 cell right here. Okay? Can develop into different subsets. Memory covered. Okay, good. TH2, TH1, TH17, T regulatory. I'm going to tell you what you do not have to know. So I don't really care about TH1 and TH2 subsets. Okay, you don't have to memorize which one does what. What I want you to understand is that, look, CD4 cells, what type of MHC molecule do they recognize? Class 2. By the way, I created my own mnemonic rule, how to remember which one. I call it, it's always 8. Type 2 is recognized by CD4. 2 by 4 equals 8. Type 1 is recognized by CD8. 1 by 8 equals 8. Just, it's a stupid rule, but usually stupid rules, they memorize the best. Okay, so we know that CD4 cells recognize MHC class 2. Which cells, except dendritic, have MHC class 2 on their surface? Two other types. Macrophages and B cells. Let's talk about B cells. So CD4 cell recognizes MHC class 2 on the surface of B cell. What is the function of B cell? To become plasma cell and produce antibodies. CD4 cell makes this process way more efficient. Does that make sense? Macrophage. What's the function of macrophage? Phagocytosis. CD4 cell interacts with MHC class 2 of macrophage and makes the phagocytic activity way more efficient. Does that make sense? Moreover, CD4 cell produces chemicals, cytokines, that stimulate maturation of CD8 cell. So it helps CD8 cells as well. Okay, now, before we move on to other couple of types of T cells, I usually compare CD4s, mature CD4s, T helper cells, to a football coach. Okay? When you look at the football game, football coaches, they don't go on the field. They don't play necessarily, right? It's a quarterback, it's linebackers that are in the field, right? Does that make sense? But coach tells them what to do. Can the team play without the coach? Yeah, look at New Orleans Saints 4 years ago. They did. They finished the season 8-8, and it was a miracle that they did 8-8. It's awful they didn't have a head coach. Okay? So, will macrophages phagocytose without CD4 cells? Yes, but not as good. Will B cells produce antibodies without CD4s? Yes, but not as good. Does that make sense? Will CD8 cells mature? Yes, but not as good. CD4 cells make your immune system extremely efficient. Is that understood? Now, little mention, Th17 cells have specific function to just stimulate inflammatory responses. And there's another type of cells that becomes more and more, well, its importance now is very well appreciated. It's T-regulatory cells, or Tregs. These cells, they dampen immune response. Okay? They slow it down, they inhibit it. Why? Why? Huh? Exactly. Bingo. Yes, it's time to shut it off. You know, if your immune system keeps going, it will start to attack your own tissue. There's a likelihood, right? So, and it was shown that in autoimmune diseases, there were many studies, especially regarding inflammatory bowel disease. I was exposed to it, and I am exposed to it now. My very, very good friend studies inflammatory bowel disease. That number of T-regulatory cells in the inflamed intestinal tissue, inflamed intestinal mucosa, is very much decreased compared to a healthy one. So it looks like Tregs, when they are absent or not coming into the tissue, they somehow influence. And they show that if you increase the number of Tregs in the inflamed mucosal layer, of the intestines that decreases the symptoms, ameliorates IBD. The only problem is that you don't really want to mess with immunity. Okay, CD8 cells. CD8 cells recognize what type of MHC? Class 1. So they are presented, so antigen is presented by type 1 MHC. Remember, type 1 MHC, there is one on antigen presenting cells, so it's fine. Okay, and then CD8 cells differentiate into, of course, the pool of memory cells, and they differentiate into the CTL, cytotoxic lymphocytes. And these guys are killers. It's like in a Hollywood movie, you know, some mafioso boss shows the picture of a next victim to the killer, and killer just goes and looks for the victim. Does that make sense? So those CTL, cytotoxic lymphocytes, they go into the circulation and go around the human body, and if they spot, if they recognize the cell with the same antigen that was presented to them, they kill it. And remember, they recognize antigen presented by which type of MHC? One which is present on all nucleated cells, so they can recognize that precise particular antigen on hepatocyte, fibroblast, splenocyte, epithelial cell, muscle cell, whatever. Does that make sense? Now, how do they kill it? They do not phagocytose. We have two cell types, which way of killing pathogens is phagocytosis. They are macrophages and kamikazes, neutrophils. Okay, those two. CD8 don't phagocytose. What it does instead, one way is Fas ligand, Fas receptor. That's your CTL, that's infected cell. It's kind of huge ligand. I don't remember which one has what, but it's an idea. Okay. When there is an interaction like that, receptor and ligand, it triggers the apoptosis of the infected cell. Does that make sense? Second way is granzymes and perforins. Perforins, surprisingly, perforate. Okay. And granzymes, they enter through the pore that is produced by perforin. And granzymes stimulate apoptosis. They trigger apoptotic response in the cell. Does that make sense? Now, if you think about this, CD8 cells, what type of pathogen that would be more effective? Extracellular or intracellular? Intracellular, absolutely, yes, of course intracellular. They directly kill the infected cell. Well, which obligate intracellular pathogens, you know? Like, they all intracellular. Viruses, yes. Well, guess what practically all viruses love to inhibit? Which normal cellular process? It's pretty simple question. I told you the name of that process like 30 seconds ago, but you have to make a leap of logic. Think about what CD8 cells do to the infected cells. Bingo! Viruses prevent apoptosis. You look at almost all virus infection, they do it by different mechanisms, but one of the virus's effects, they prevent apoptosis. Not forever, no. They just lead to the survival. There was an experiment, very simple experiment, that a colleague of mine, well, I call him colleague of mine, he's like distinguished professor. What they did in the lab, they took monocytes, and monocytes can be easily infected by cytomegalovirus. So they took two pools of monocytes. One was uninfected, primary monocytes. They isolated them from the blood. That was one of the donors. So they took a pool of uninfected monocytes, and they took another pool and infected this pool with cytomegalovirus. And they looked at the survival of cells. Uninfected monocytes died like 4 days later. Just normally, I mean, cells are not suited to survive for that long in the flask. They went into apoptosis. Infected monocytes survived for 14 days because virus prevented apoptosis. The only reason to replicate. Enough time to replicate, get out of the cell, infect something else. Does that make sense? So viruses are pretty good at inhibiting apoptosis. Got it? So before we move on, understand, I want you to have a good grasp on differences between MHC class 1 and 2, where you can find them. How do they present antigen? Okay, so class 1 presents antigen synthesized in the cell. Class 2 presents the products of pathogen breakdown. Which cells are presented by, so MHC1 presents to CD8, MHC2 presents to CD4. So understand the concept of MHC restriction. Okay. Understand the process of activation, which is rather simple. Functions of CD8: B cell activation, macrophage activation, CD8 cell activation. Okay. Process how CD8 cells kill other cells, infected cells. Right? It's all in the study guide. Any questions? Moving on. That's kind of a summary. If you want, we can go over it. So it gives you, pretty much tells you the quick summary of what I just told you to know. Okay, where it is, where you can find those MHC molecules. Okay, what are they recognized by? What type of foreign antigens endogenous, that are synthesized in the cell, or exogenous products of phagocytosis, and funny explanations of the functions. Okay. So this is a very bold attempt to summarize in one slide the entire human immune system. Okay, it's sort of an overview of what we were talking about up to this point. You have 3 defenses, intrinsic defenses: dry skin, acidic stomach, mucus and cilia in the upper respiratory tract. Okay. Understand the locations, understand what's going on in each location, right? You have innate immunity, okay? We talked about them. Cells, white blood cells like neutrophils and basophils and eosinophils, macrophages, complement, interferons. Those are components of innate immunity. And then you have adaptive defenses. Okay? The B cell response, the humoral response, does not require the presentation. B cells, they produce memory cells, and they differentiate into the plasma cells that release antibodies. Okay, now, antibodies. Quick thought to you. Antibodies, can they opsonize? Yes, they can. Now, what is the purpose of opsonization? Yes, to make the cells more susceptible to phagocytosis. I couldn't say it better. Okay, so you have link between humoral response and innate, which is phagocytosis. Does that make sense? You look at the T cells, they have to be presented with antigen. So antigen presenting cells do it. Then CD4 cells will develop into the helper cells that will either stimulate the differentiation of B cells and antibody production, or and stimulate phagocytosis, and will indirectly, not through the direct cell-cell interaction, will stimulate the T cell activation. Sorry, CD8 cell activation. Now, CD8 cells will kill infected cells. So here you have a great link between the cellular and humoral response, the CD4 and B cells. You have a link between, another link between adaptive and innate, between the cellular immunity and phagocytosis, okay, CD4 macrophages, right? So they all linked. Does that make sense? Understand these links. You know what really helps? I don't know about you, what we did with at one point when people had troubles, we pictured it. We did a flow chart. So when you use your notes, just start to picture it. You don't have to be very detailed. Just show links. You know what I'm saying? Between different processes. And usually like one A4 page is enough. And you'll be pretty proficient about it. Yes, that's my favorite part. Well, I have so many favorite parts, as I mentioned yesterday. Vaccines. Arguably, well, along with the development of a toilet, it's probably one of the greatest achievements of public health in the 20th century. I'm not kidding about the toilet. Okay. So vaccines, the basic idea is that you expose your immune system to a pathogen or a fragment of a pathogen without causing an actual infection, without causing a full-blown disease. Got it? So what types of vaccines can we think about? So imagine a pathogen. For simplicity, well, we can use virus, we can use bacteria, it doesn't really matter. Let's use bacteria, okay? So what will, you have a bacteria, some kind of bug. What if you'll inject the person with a live bacteria? They got not only infected, what else? Disease, yeah, they're going to get disease, right? So what do you want to do with that bacteria to, bless you, to prevent disease from happening? Which you can do, yes. You can kill it. Okay, how can you kill bacteria? You can heat it up. Perfect, you can heat it up. Any other ways? Come on, how else can you kill bacteria? Yep, formaldehyde is really good. Formalin, actually. Because you can get rid of it later in the manufacturing. What else? UV. Well, UV is probably a little weak. You want something more powerful. But yeah, radiation. You can irradiate it. Absolutely. Perfect. So you inactivate it. I got UV here, but UV is probably not going to be enough. Okay? So you inactivate it. You have that's kind of an awkward, very awkward scheme. So the idea pretty much is you have dead bacteria or inactivated viruses, but they still have the proteins, okay? So they still have the antigens. You stick it into the patient and you get your immune response. Which inactivated vaccines do you know? Injectable, yes, injectable flu. The one that you receive in the IM. MMR is not. MMR is different. But polio, IPV, IPV is inactivated. Pneumococcus? Yes, it is an activated vaccine, yes. It's also partially purified. Okay, meningococcal, pneumococcal, Haemophilus influenza, Hib vaccine, inactivated vaccines. Right, so that's one way. Attenuated. What does that mean? Vaccine is attenuated. Pathogen is what? Weak. Yeah, it is weak. Which attenuated vaccines do you know? Oh, we don't have one. We do not have malaria vaccine. Unfortunately, no. Well, you tell me. It's viral disease. It was common on the east coast of... Huh? Yellow fever, yes. One of the most successful and most effective attenuated vaccines ever. Okay? Yellow fever virus. 17D strain. Vaccine was created by Max Tyler in 1937? 30s, okay. He passed it through monkeys and rabbits. He was alternating monkeys and rabbits 200 times and got attenuated. What does it tell you about attenuation process? How well do we know the mechanisms? We do not know anything about mechanisms. It's all trial and error. Let's do this and see if it works. Okay, let's pass through this and see if it works. Okay, Pasteur did his rabies vaccine, attenuated rabies vaccine, passed it through rabbits like crazy number of times. Okay, well, yellow fever is not that common. By the way, according to the current clinical standards, if 17D vaccine against yellow fever would be developed now, it would never get approved by FDA. Because if you inject it into mice, intracranially in the brain, it causes paralysis. I mean, subcutaneously it's fine. In humans it's fine. There were 2 or 3 cases of side effects recorded for 17D. It is safe, extremely effective. Okay. What else? Attenuated vaccines. Mallory mentioned one. MMR, attenuated, was developed by Maurice Hilleman, American microbiologist. And actually the mumps strain that is used in the vaccine was isolated from his daughter. His daughter got mumps and he used this virus to attenuate it. Oral polio vaccine. It is not used in the United States. It is used in developing countries. Okay? It is... So what is the biggest concern with all attenuated vaccines? What do you think? Yes. Well, they test it, so by the time you inject it, you know that it's weak enough, but what if it gets in the person with, Say again? Yes. Yes, immunosuppression. A great example is the smallpox vaccination. First of all, we do not have smallpox, but US military, they still vaccinated against smallpox. And there was a case when a recruit was vaccinated against smallpox, and he had undiagnosed leukemia, and he died. So it wasn't like a malpractice or anything. He died because nobody knew that he had leukemia. And smallpox vaccine is attenuated vaccine, right? Same goes for things like, there was a quite an interesting case. A soldier got vaccinated against smallpox, came to visit his girlfriend, and they had sex, and the vaccine was transferred in scarification, so it gets rubbed into the skin. The virus got transferred to the mucous membranes, vaginal mucous membranes, and she developed actually the focal infection, the mucosal infection, with that vaccine virus, because it wasn't in the right place. She wasn't immunocompromised, but it wasn't in the right place. I mean, she was treated and everything was fine, but it was kind of weird. Okay. Now, there is an approach to use parts of the pathogen. So, the subunit vaccine is illustrated here. Okay. So you don't take the entire cell, okay, or you don't take the entire virus particle. Instead, you produce a part of it, which is one of the best vaccines that we have. It's pretty expensive, but it works perfect. That is subunit. Extremely safe. Kids usually get it like first day of their life. Hep B, yes. Hep B is a subunit vaccine. Very effective. Very safe. It's pretty much, I mean, it's a protein. Nothing can happen. Really, nothing. The main side effect of the vaccine is redness in the point of injection. That's kind of a tolerable, side effect. So the problem is that the manufacturing process is kind of expensive. Okay. Vector vaccines, they're not in the picture, but think about this. If you have yellow fever virus, right? You have a very safe vaccine. What if you take the guts out of yellow fever? Bless you. Some of them, some genes out of that vaccine virus, and put there the genes from the virus that's related to it, like West Nile virus or Japanese encephalitis. This is called chimeric virus. Okay, it's like trying to put some monkey genes in the humans. The only difference is that in monkeys and humans it's not going to work. In viruses it works pretty well. So you can take a couple of genes of yellow fever and substitute them with a couple of genes for Japanese encephalitis. And we now have such chimeric vaccine. It's called Chimerivax, produced by Sanofi Pasteur. It is used for vaccination against Japanese encephalitis, and yellow fever, the 17D strain, serves as a vector. Does that make sense? In the wake of Ebola epidemic, first of all, we didn't have Ebola vaccine at the time epidemic started. Why? 2 words. Nobody cares. It's in Africa. That's it. That's all explanation. There's no market. And the number of people that acquire Ebola is not that huge, to be honest. So, but when vaccination was, when the outbreak was almost done, the pharmaceutical companies, nevertheless, they rolled out the vaccine, and it was a classical vector vaccine. It was attenuated so-called vesicular stomatitis virus, VSV, which carried one of the protein genes, which carried a gene for one of the Ebola proteins. And, well, the main problem is that the outbreak was almost over, so when you have very few cases, you cannot really test how effective the vaccine is. Well, they did the clinical trials anyway. It was well tolerated. there were practically no side effects except for redness in the site of injection, and they showed that based on the data that they had, it was pretty effective. So next time there is an outbreak, and there will be, they can do so-called ring vaccination. So if they identify a person with Ebola, they vaccinate all contacts to limit the spread. So instead of constantly vaccinating every person in the region, which is not feasible and not practical, they do this. Does that make sense? Now, one thing before we move on to the next slide, which I want to discuss with you, are vaccine development strategies still trial and error? Absolutely. So we have, if you read the papers about vaccine development, they implicate all types of immunological mechanisms. Oh, this is why it's going to work like this, and then bam, it doesn't. Okay? So it's trial. We hope. We have the idea why it should, but when it doesn't, we say, whatever. Well, it didn't work this time. We got to try something else. Great example is HIV vaccine, which doesn't exist. And we tried everything from inactivated HIV to attenuated HIV to subunit vaccine to now there is actually a big study in different centers going on. There's a guy from Oregon State Health University, I think that's the name of the college, or University of Health Sciences, whatever. He developed a vaccine which is a vector vaccine using cytomegalovirus as the vector. So he put some HIV genes in the CMV, and it showed surprising efficacy in monkeys. So they now, and he invited all, other researchers to come and check. Maybe it's a mistake, and they were going over it for like 2 or 3 years by now, trying to figure out, is it going to work? Okay, maybe we will have one. So, questions. This is what, if you go into nursing, part of the nursing that, I know some of you will not work with people that can talk. Like in dental hygiene, you have an advantage. A person, for their own safety, they shouldn't talk. Okay. In other nursing professions, I don't know, if you work in intensive care units, they might be a little weak, or vaccines is the last thing that worries them. Okay. But if you encounter regular patients, if you are in internal medicine, pediatrics, you may have people, that will ask you, will question vaccination. There are 2 types of people who question the vaccines. One type is people that are genuinely confused. So they need just clear, logical explanation. Unfortunately, that's a minority, according to my observations. Other people that think that vaccines is a government conspiracy. Well, what can I say? It's really hard. And they will give you 1,000 anecdotal cases, And you have to understand another concept before we move on. There's a fancy Latin saying, post hoc ergo propter hoc, which means if one thing happens after another, then there's a causative link. Pretty much means if I leave home without the umbrella and it starts raining, it rains because I don't have an umbrella. Okay? We all know that rain starts because you washed your car. Things like that. Which is obviously, not truly a link, but if a person vaccinates a child and then something bad happens, they say, oh, it's because of the vaccine. I'm not saying that they don't have side effects, and we'll get to that, but you have to understand it's a little bit trickier than that. And in medicine, you cannot work with single cases. Medicine is the science of big numbers. So, why do we vaccinate against diphtheria, pertussis, rubella, measles, mumps, polio? I just named 6 diseases that, except for maybe measles, other diseases, I bet, well, pertussis also. Other than that, I believe there are physicians that never saw any of those, ever saw any patients with those diseases. Why we still, what is the point? I mean, maybe we should just stop. Exactly. And it's kind of funny to look at the vaccination in this way. Vaccinate people against pertussis. It's a DTaP, diphtheria, tetanus, acellular pertussis. Vaccine has pretty decent side effects, actually, especially in kids. Kids often develop headaches, or when they're too young to report headaches, they just cry all night, they may run a fever. So it's pretty reactogenic vaccine. Sounds lousy. And we don't have any diphtheria, we don't have any pertussis around. So it kind of looks this way. Vaccine causes a hell of side effects, and there's no disease. Well, as you know, you get rid of vaccine, you get no side effects, and immediately a lot of disease. Measles is a wonderful example for that. Okay, so we keep it away. Example: Nigeria, Afghanistan, Pakistan. Afghanistan, failed state. Practically no polio vaccination. They have wild polio cases. Nigeria, at some point, Nigerian government decided that through polio, WHO, led by Americans, want to establish the power, the control over the country. Well, it's a lousy way, I would say, through vaccination. So they banned vaccination in Nigeria. 3 years later, when they had an epidemic, they realized they should let them back. They still try to eradicate polio. Okay? Pakistan, it's not the entire country, northern provinces that border Afghanistan. So get rid of vaccines, you have the disease coming back right away. Okay? I really like the story about Oregon, where you have a bunch of wealthy, educated people that, no offense, understand me right, but people who shop at Whole Foods and drive Priuses, they also don't vaccinate their kids because they believe in the natural way. So there were outbreaks of measles and mumps in that particular locale. Okay. So herd immunity. You can say, my child is fine. My child is healthy. Okay. I don't want to vaccinate him. And you tell them, we need to provide herd immunity. And they say, I don't want to be a part of the herd. I don't care about other people. Which is fine. I understand that attitude. Okay. What is herd immunity? Is every person, say again? Yes. Can every person receive vaccine? No. Who cannot? Immunocompromised people with allergies, right? Some people have allergies to almost everything, so they just cannot get any shots because they're allergic to various components of the vaccine. Okay? So they cannot receive a vaccine. Okay? What protects them? Herd immunity? Well, viral diseases, good luck with that. Measles, mumps, rubella, polio. No, there are no drugs against those diseases. So the only thing that protects them is the herd immunity. Okay? We got to get more than 95% coverage. In this sense, vaccine against flu is an example of altruistic vaccine. You're probably going to be fine. I mean, it's going to be a miserable week when you have a flu, but other than that, you're going to be fine. But if you vaccinate, you break the transmission cycle. You decrease the chances of other people to acquire flu, to get it from you. and we talk about immunocompromised, we talk about elderly, we talk about hospital patients. Does that make sense? For who flu can be quite deadly. How safe? Vaccines are safe. Much safer than to get the disease. Don't get me wrong, there are side effects. They are all reported. Absolutely. For instance, oral polio vaccine has a side effect, so-called vaccine-associated, vaccine-derived polio. I'm not going to get into the biological details, but you know how frequent that side effect is? 1 per 3 million vaccinated. You probably have higher chances to get in a car crash. Seriously. So, vaccinate. That's my favorite question. Infection is the natural course is better. Let nature take its course. I always say to people, oh, you're all for natural selection? Get ready, your child isn't getting selected. You know? That's sometimes gets them sober. Okay. Great argument, by the way, for anti-vaxxers. Tell them not to take tetanus shot next time they step on the rusty nail. That's going to be thrilling, right? Or a dog bite. That's going to be even more thrilling. There is a chance to survive tetanus. There's no chance to survive rabies. And the clinical picture is colorful. Okay, especially in the, I think it's called furious rabies, the one that develops rapidly. It has all kinds of wonderful things like hydrophobia and photophobia. Side effects, all of them reported. If they severe, vaccine usage is paused. Example is the first version of tetanus, diphtheria, pertussis. Initially, it had pretty much inactivated Bordetella pertussis microbes in it. And it caused pretty bad side effects, like people ran fever for a couple of days. So it was very active, but also reactogenic. What they did, they cleaned up, they identified that it was a pertussis component, right? And they got rid of a lot of non-immunological things, not things that are needed for the immunity. I don't remember what they got rid of. It's called a Cellular, so kind of cleaned it up, okay? Not surprisingly, the efficiency also went down a little bit. So you now need several boosters, okay, of vaccine to give you a proper immune response. Do they cause autism? Andrew Wakefield in 1995 published a paper, in 1997 published a paper based on the study of 12 children. 5 of them were fake. They never existed. That there's an association between the MMR vaccine and autism. Since then, his findings were debunked by a gigantic meta-study by Institute of Medicine in the United States, which based its meta-analysis on more than 1 million vaccinated kids, finding absolutely no statistical evidence of the link between autism and vaccination against MMR, polio, and I don't know if they study any other vaccines. Right? Andrew Wakefield, his medical license was canceled. He lives in Austin, Texas, and continues to perpetuate the idea that vaccines cause autism. His conflicts of interest included the funding from law firms, and the specialization of those law firms was to sue pharmaceutical companies. So there was 100% conflict of interest. Everything was debunked. They even fired Jenny McCarthy from the TV. There is a website called Jenny McCarthy Body Count, by the way, which statistically estimates how many people died from vaccine preventable diseases in the United States. And since the moment Jenny McCarthy started the program, started to propagate the idea, vaccines cause autism, she has an autistic child. And she claims that autism was caused by vaccine. So she started that propaganda. Since then, about 1,000 people died from vaccine-preventable diseases in the United States because they were not vaccinated. Tens of thousands got sick. Recently, analysis demonstrated that her child's autism is genetic. Has nothing to do with the vaccine. Now, people ask you how vaccines cause autism. There's a great website that explains how they do it. Okay, I think I need to switch it. Okay, yeah. Give it a second. Computer is rather slow. Okay, you got your explanation, right? That's the link to research. The American Association Academy of Pediatrics. Okay, it really, when I see anti-vaxxers, it makes me crazy. Okay, so we got this. Big Pharma. Do they make money off the vaccines? Yes. Oh, don't get me wrong. Pharmaceutical companies, they not like this charity funds. No, they want to make. It's not their exactly. Oh, absolutely not. Because look, you can charge a ton for vaccine. Because if you want the public company, look, who gets vaccinated? If you have, I don't know, $1 million a month income, you're going to get your kids vaccinated, and if the vaccine costs $200, even with the insurance, it's not a problem. Okay? Now, if your monthly income is $10,000, okay, and vaccine costs $200, then it becomes kind of a problem. So vaccines cannot be expensive. Usually, state will cover part of the cost or the entire cost. Okay, so it's state-sponsored programs, and government doesn't want to pay a fortune. Okay, so they cannot be expensive. Right? That's problem number one. They have to be, and if they cannot be expensive, they have to be distributed in large amounts. So we talk about millions of people. For instance, when I did PhD, I worked in a facility that made vaccines, including yellow fever vaccine. It was wonderful. We had a perfect client, military. Every year, as mandated by law, they had to buy 200,000 doses. We tried to establish the study to show that vaccine can be stored not for a year, but like for 5 years. And a supervisor told us, "Are you nuts? Keep it to yourself. We want them to order every year because it is expired. Just keep going. We know that it's fine, but just keep going. Just keep going." Okay, so that's fine, Military ordering, state ordering is fine, but 200,000 is not a lot. If you talk about polio, there are millions of doses. Polio vaccination, by the way, oral polio vaccination, I think it costs 1 cent in Africa. So it's really cheap. So nobody's going to make vaccine against Ebola. Right? It's a small market. My kids are vaccinated now that's my personal point. First, my kids are vaccinated. Every vaccine, even not mandated. Like HPV is not mandatory vaccine. My kids have HPV shots. Okay. They vaccinated against flu every year. That's entirely altruistic. They can withstand a poke in the shoulder. That's fine, but other kids will benefit. I do not want them to play with unvaccinated child. Why? What if vaccine did not work in them? That happens. Vaccines are not 100% effective. They're 95, 99% effective. Some of them, like mumps, it's about 85, so you got to get boosters, okay? What if they were unlucky, and particularly in this case, they didn't get high enough titers of antibodies or good T-cell response? I don't want them to be exposed anyway. I don't want them to play with unvaccinated child. I know they are protected. If, I don't know, if I go into the building site, I'm going to wear a hard hat. But it doesn't mean that I'm going to scream to people, hey, throw bricks at me and see if it's going to hit me or not. Okay? No. Second, I am 100% for vaccination for public schools. No philosophical, no religious, all that bullshit exceptions. No. If you don't want to vaccinate, there are private schools all over the place. And I'm really happy that New York and California finally got to this point. In not state of New York, but city of New York, there are no exceptions whatsoever. I love the way it is done in Australia. If you do not vaccinate your child, you lose your tax incentives. That's it. Tax incentives. Tax breaks. That's it. You just pay, and as far as I know, it's pretty good hike. Tax hike. So it's up to you, because public health system will, and it's government-sponsored public health. Government has to pay for all the complications from not vaccinating the child. I know that a lot of doctors, physicians in the United States, they refuse to admit, to kind of work with a family with unvaccinated kids. Their reasoning is perfect, and it's true. I have all kinds of kids that come into my practice, and I cannot allow your unvaccinated child to potentially transmit the disease to a kid who has, I don't know, a cancer. Does that make sense? So that's great excuse. It's not an excuse, it's a reason, actually. Okay? And last but not least, with many people who oppose vaccination, the reason doesn't work. So you give them logic and they tell you, I washed my car and it started raining. Okay? So if you know anecdotal evidences, if you know anecdotes, tell them, I have mine. I told you before, my wife's friend, her son died at the age of 1 and change because of measles between 2 vaccinations. So the person fully protected after 2 or 3. He didn't get his full protection. That is. Okay? And have your anecdotes. Right? Yeah, I think I finished with my rant about vaccines. We can take a break.